Please welcome Shikma Litmanovitz, Technion, Israel Institute of Technology.
My name is Shikma Litmanovitz, Director of Business Development at T3 Technion Technology Transfer. My prime mission is to continue crossing boundaries and creating bridges between academia and industry. Today, I have the pleasure to nominate Mana.Bio as the Science Breakthrough of the year 2023. What is Mana.Bio distinguished for? Mana.Bio, founded by Professor Avi Schroeder from the Technion Warsaw Faculty of Chemical Engineering and Professor Kira Radinsky from the Technion Department of Computer Science, is an AI-based drug delivery startup. Its groundbreaking technology focuses on creating a platform for lipid nanoparticles development for oligonucleotide therapies, including DNA and RNA-based therapeutics, vaccines, and gene therapy.
Breaking the wall to unlocking RNA delivery through AI. Yogev Debbi, Mana.Bio.
Hello, everyone. I'm happy to be here, calling in from Tel Aviv, Israel. I am co-founder and CEO at Mana.Bio, and we are unlocking the RNA therapy. RNA is the next evolution of therapeutics and vaccines. And as you can see, this is a huge market, not only in terms of market size potential, but also in terms of the impact on human health. You can see all these diseases are just sample of diseases that we already know the mechanism of action, we know the biology, and we know that we can solve them with RNA. However, one of the biggest challenges is how you deliver RNA into the human cell. And in fact, Jennifer Doudna, the CRISPR Nobel Prize, said that this is one of the challenges with CRISPR, the delivery. So what's so challenging with the delivery? Imagine an astronaut that needs to get to the space station to fix it. You really need a space shuttle with oxygen, seatbelt, pass the atmosphere, land safely. You cannot just walk there. So this is kind of similar to RNA. You need to keep it stable. You need to take it to the right cell or tissue, and you need to be able to pass the cell membrane. So the underlying logic of how to do it is really unknown. This is why many companies in the space that are working in the space of delivery, they use this trial and error cycle of trying to figure out what will work and what will not work. And this is really a lot of time and money in this process. So our approach is this engineering mindset of design, build, test, learn cycle, where we design candidates by the AI, we then build and test in our lab, and the important part is learn, where every experiment result go back to the database, make the AI smarter and better. And we leverage multiple data sources for our AI. So one is literature. We developed automated software tools that go to existing databases such as Google Scholar and take all the patents, publications, research papers, and we developed software bots that go over the actual PDF of these papers and take the text, the images, the charts, and everything go automatically to our databases of the training set of the AI. We also leverage a high throughput screening mechanism in our lab, generating hundreds of data points every week. Using this approach, we already discovered multiple well-curated IP-protected delivery of RNA, all validated in vivo. So just in a nutshell, under 2 years since inception, we were able to take this technology from concept to existing platform. So we started from pre-seed of $5 million as a pre-seed raise. Then we moved into predicting in retrospect of commercially available delivery systems. Then we moved into the first AI-generated delivery of RNA into liver. And then we moved into targeted delivery, and we started with lung. We took that to optimization level, and after doing lung, we started with other tissues such as kidney. And using these steps and these milestones, we just recently closed an oversubscribed seed round of $14.5 additional million by lead biotech VCs. So to sum up, RNA therapeutics is the next revolution of pharmaceuticals, and delivery is the key to unlock it. Under 2 years, we were able to de-risk the technology. We created an IP portfolio with 2 patents pending. And we started engaging with pharma companies to prove our business model. And our business model is collaborating and partner with pharma companies and license our tailored delivery solutions and enjoy milestones and royalties once the drug is in the market. Lastly, I'm so proud of our team. The founding team consists of Professor Avi Schroeder from the Technion, chemical engineer, a drug delivery expert. He is also an alumni of Bob Langer's lab at MIT. Kira Radinsky, she's a machine learning expert and serial entrepreneur. And Roy Nivo and myself, we go back 20 years together, actually. Our background is software engineering. We started our first biotech company a decade ago, sold it to Twist, and then IPO'd. And our advisory board consists of veterans and experts in different fields, such as medical, science, and strategy. Thank you very much for listening.
Great. Great, that's great. I hope you can hear me well. And then we're going to take the questions right now. So this is even more important that we wait for the microphone here. We're going to start with you, Chaz and Frank. We will have Angelica. It's the usual suspects here, right? Let's go. Thank you.
Yes. Thank you very much for your nice presentation and nice data about the lung-specific delivery. Ask myself, have you repeated this data with LNPs with different payloads? That means different lengths of inserts, of RNA insert, different charge, maybe even totally different payloads beside RNA, because as far as I know, LNP and the payload makes the drug who gives the tissue sensitivity.
Yes, thank you, definitely. This is why we think such a platform is so important, because the LNP depends on the payload, depends on the tissue target, depends on the administration route. So this is a super valid question. So far, we worked only with reporter payloads, so mRNA and mCherry, and definitely other payloads will have to be optimized by the LNP. I have to say that we do have models for other payloads such as siRNA as well. And the next step for us is to validate in vivo different payloads, as also CRISPR and DNA. Thank you.
Okay, next question by Chaz, please.
I mean, you've clearly demonstrated effects in liver and lung and kidney and brain. Do you think you could increase the value of the company much more quickly if you just took an asset for liver all the way to the clinic?
Oh, liver! This is the interesting thing. I thought the question is going to be take something to the clinic, which is obviously will increase the value of the company. Liver specifically might. I mean, currently it's a bit, there are multiple projects for liver. So for liver specifically, it's a good question. I will broaden the question. I think that we will increase the value of the company once we develop our own pipeline. However, started 2 years ago, the team is mostly focused on R&D. I don't think at this point we are tuned to start our own pipeline. So we decided to start with the coming 2-3 years of collaborations where we out-license our delivery solutions. In that time, we will earn 3 things. One will be cash, obviously. The other would be validation to our technology. And the third one will be education to our team. And at that point, we will develop our internal pipeline, perhaps also for liver solutions.
Okay. Next question by Angelica, please.
Yeah, thank you very much. It's a very important field that you worked on. Yeah. But it's also a very competitive one. Yeah. Did you have checked if you have freedom to operate by your filed patents?
Yes, absolutely. So the field is competitive. Most of the competition, as I said, are doing what we call real science. So it's a lot of trial and error, and we come with the AI, and we're able to achieve faster results. The patent landscape is really crowded. You're absolutely correct. This is why when we just started and we got this feedback, we built a database that consists of all the patents that are out there. And we have ways with AI to compare structures of molecules. So we automatically, as part of the process, we compare the structure of what we want to discover to everything else that exists there. If we see it's too close, then we took it off. All the patents that we filed, we proved in our systems that are that are different enough that everything else that is out there, and we pursue with the patent. So we have this ability as well. Thank you.
And this is wonderful because it is 6 seconds left. We won't make it to the next question. Thank you so much to have been virtually with us today. And thanks for the questions. And we're going to move on to the next pitch. And this time it will